All right, let's begin unit number 6. We'll be diving in on the next system, our cardiovascular system, where we talk about 3 interrelated components, as it states there in the first sentence. The blood is obviously one of them, what we'll talk about in this lecture. But then we've also got 2 other components, are blood vessels and the heart. Now, the very next lecture after we finish this one, which I said it's only 5 slides, so we should be able to get through this without too much trouble. We'll kind of take a detour away from cardiovascular system and dive in with the lymphatic system, which there's a lot of overlap between the 2, which is why I talk about the blood and lymphatic system. You'll kind of see why as we move on into that lecture. So we'll kind of get into that later here this morning. Okay, so like I said, 3 components of our cardiovascular system include the blood, the heart, and our blood vessels, which function to really kind of get our cells the nutrients that they need and remove the waste that they produce, because most cells of the human body cannot move around to obtain their O2 and nutrients or eliminate CO2 and other wastes. Instead, we use our blood, which is circulated through our blood vessels and pumped by way of our hearts. It's not the first time we've talked about blood. We learned about blood just briefly back in our tissues lecture. We learned that it's a liquid connective tissue composed of a liquid cellular matrix called plasma, and that dissolves our cells and cell fragments, what we call the formed elements, which includes red blood cells, white blood cells, and platelets. Very closely associated with our blood is interstitial fluid, fluid that bathes body cells and is formed when fluid diffuses from our blood and travels out of it and between our cells, because that's where you find interstitial fluid. Remember, inter means between. So that's fluid between our cells, which can be taken up by our cells, becoming intracellular fluid. It then is not going to stay stagnant and stay in our cells. It'll leave and become interstitial fluid again, and then we'll return back to the blood. And this is just a cycle of events that is always occurring. Once again, demonstrating that fluids in our body are operating like a river, circulating and not staying stagnant. And 3 just general functions of our blood are listed here at the bottom, with examples demonstrating those different functions. First is transportation. Blood carries oxygen from our lungs to all the body's tissues. Carbon dioxide is carried from those tissues to the lungs. We pick up nutrients from our digestive tract, things like glucose and other carbohydrates, amino acids, some fats, water, electrolytes, a lot of different things that we ingest. Metabolic wastes are carried to the kidneys to then be filtered out and made into urine to be excreted out of the body. We just recently got done talking about hormones and how we carry those to our target cells. We transport stem cells from bone marrow to tissues. Our blood provides us with various forms of protection. It's involved in a process called inflammation. Our white blood cells provide immunity. We'll talk about the 5 different white blood cells and what specifically they do for us. Our antibodies produced by white blood cells help neutralize toxins and pathogens. Pathogens is just a fancy word for anything that is a disease agent, disease-causing agents. Platelets initiate blood clotting, which helps us to keep from losing all our blood when we get cuts and that kind of stuff. Blood regulates different things like our fluid movement across capillaries to distribute and stabilize fluid distribution. Our proteins in the blood act to buffer and stabilize pH of our body. And we use our blood to regulate body temperature by transporting blood to our skin or visceral organs, which is something we do when we're hot or when we're cold. For example, when we get really hot, we transport more of that blood to our skin, which is why places like our face get, red in color. We're doing so to try to, cool that blood by, getting it in closer contact with, the sweat that's on the surface of our skin. We can fan and move air across the surface of our skin to help cool the blood. And then visceral organs is what we, we'll move the blood to, like when we are really cold, kind of keeping it internally so that we don't lose as much of that heat to the external environment. Down here at the picture, or down here at the bottom is a picture that we've actually seen before. It's that same picture that was in that tissues lecture when we first introduced blood. It is a view of a sample of blood under a microscope zoomed in 400 times, showing you the majority of our cells are the are the red blood cells or erythrocytes that we'll talk about here in a couple slides. Then you've also got various forms of white blood cells shown here, kind of the bigger, blobbier looking cells. The little speckles here, kind of sprinkled in, are the platelets, which are not actually cells. We'll talk about what those are here in a little bit. And then the space in between those blood cells would be your plasma, which we'll start kind of talking about here on this page. Now that picture is not important. There won't be a single picture from this lecture that will show up on your exam. So your first 20 questions, even though there's only 5 slides on this lecture, there will be 20 questions because there's still quite a bit of information. But those first 20 questions that are on exam 6, there won't be a single picture. All right, so just kind of reading through here. Blood is described as more dense or denser and more viscous. Viscous refers to the thickness of a fluid. In this case, more viscous means it's thicker, and we're comparing it to water, so it's more thick or thicker than water. Has a slight sticky feel to it. Temperature is about 38 degrees Celsius or 100.4 degrees Fahrenheit, which is about a degree Celsius higher than oral or rectal body temperature. Has a slight alkaline or basic pH ranging from 7.35 to 7.45. 7.0 is neutral. That would be like what pure water is. Anything above that would be a base. And so 7.4, which is right smack dab in the middle of that range, is a base, but that's a really, really weak base. So it's very, very subtle base. Total amount in young healthy females about 4 to 5 liters. So about 2 of those, kind of 2 pop bottle, if you think about them in that form. Men have about 5 to 6 liters, so about 2 to 3 of those, 2.5 to 3 of those. Represents about 8% of our total body mass, as you can see here in our kind of figure at the bottom. 2 major parts are in our whole blood samples: plasma, which represents the clear extracellular matrix or fluid portion of our blood, and that constitutes over half of our blood, about 55%. Okay. Of that, the majority of it is water, about 91.5%. Obviously, that's very, very easy to change based on your hydration status. The rest, about 8.5%, are various solutes. Primarily, we'll talk about these 3 proteins, what we call the plasma proteins: albumins, globulins, and fibrinogen. But then you've also got some other things that I'll talk about here after I get through these. Okay, let's talk about the first. Albumins produced by our liver and function as transport proteins. So they are like an Uber in our blood, carrying things like steroid hormones and fatty acids. Globulins, also produced by our liver and by plasma cells, which are a form of cell formed from white blood cells, developed from the type of white blood cells called B lymphocytes. There's 3 different types of globulins: alphas, betas, and gammas. Alphas and betas are also transporters of our iron, lipid, and fat-soluble vitamins, whereas gamma globulins are the ones that function as antibodies in our blood. So these are the ones that help us, neutralize toxins and viruses and that kind of stuff. Another term for antibodies are immunoglobulins, because they're immune in function, and they are, as you can see, globulin type. So that's another term for antibodies. Fibrinogen is our final major plasma protein produced by our liver as well, but very important, playing a role in blood clotting. For those in phys, you know what I'm talking about. I'm not going to get into all that any more than that. Other plasma proteins include enzymes, hormones, and various clotting factors, only representing about 1% of our proteins. And then the rest of our substances in the blood, about 1.5%, includes various electrolytes: sodium, potassium, calcium, magnesium, chlorine, bicarbonate, sulfate, and hydrogen phosphate. Nutrients like glucose and other carbohydrates, amino acids, hopefully not too much, lipids or cholesterol, but we all should have some in our blood. Gases include carbon dioxide, oxygen, and nitrogen. And then waste products, including urea from the breakdown of proteins, creatinine from the breakdown of creatine phosphate, which is something that we have in our muscles, uric acid, which comes from the breakdown of DNA and RNA, which are our nucleic acids, and bilirubin, which is from the breakdown of hemoglobin. So if you're not functioning correctly with regards to your kidneys, you can end up becoming, kind of toxic in your blood, because you won't get these out, because most of these are going to be excreted out in our urine. And conditions that can develop when we're not getting enough stuff out, or when organs are failing, could be things like gout. Jaundice occurs when we are getting too much bilirubin in our blood, and those are obviously not a good situation. The rest of our blood, the other 45%, are the formed elements, which are the things that have a visible structure, like what we saw on the previous page. Represent the cells or cell fragments. Includes, and by far and away the most numerous, are our red blood cells, the 5 different white blood cells, and we'll talk about what each of those are and what they do here in a couple slides, and then platelets. Now, they also have different terms. Red blood cells also go by the term erythrocytes. Another term for platelets are thrombocytes, and then the other term for white blood cells are leukocytes. Generally, I go with what's in bold here: red blood cells, platelets, and white blood cells. Just know that there are other terms for them. I like this because it just kind of gives you a breakdown on the different things and the amounts, which is really kind of handy to know. This illustration over here shows you what happens to blood when you centrifuge it, which is a process where you take a sample of blood and you put it in a machine called a centrifuge. Spins really, really, really fast and helps to separate the different parts of the blood. The blood cells separate to the bottom. Red blood cells are on the very bottom because they're the most dense. This buffy coat, the little white layer, is where the white blood cells and platelets, and then the plasma, which is the least dense, kind of separates out to the top. That would actually happen to a sample of blood if you just let it sit in a test tube for like a couple weeks. Eventually it would settle out like this, which is kind of cool. Down at the bottom is a extreme zoom in of some of those formed elements, or all of those formed elements, I should say. We're zooming in 3,500 times under a very special type of microscope, showing you kind of the shapes of those different things. Not that, it's important, but it's kind of cool to see anyway. And I always try to show you the real deal whenever possible. Like I said, none of those images and none of the images on this lecture will show up in a couple weeks. All right, what we'll do now, we'll move off of plasma, not a whole lot to talk about with that, and we'll then go into our formed elements, beginning with our red blood cells or erythrocytes. There's a couple different things down here at the bottom to fill in. Is there anything after that?
Yeah.
One more blank. There it is. Okay. Let's talk about red blood cells, which are called red blood cells because they are literally just cells that are red in color, also known, as we just mentioned, as erythrocytes, and make up about 98 to 99% of the formed elements of our blood. Two principal functions, the first of which is kind of its most important function, picking up oxygen from our lungs, delivering the oxygen to our tissues. About 98%, almost 99% of the oxygen that's transported in our blood is carried by our red blood cells. Okay. We do also pick up carbon dioxide and deliver it thanks to our red blood cells from our tissues to the lungs, but it's like 23% of our oxygen, or excuse me, of our carbon dioxide is carried by our red blood cells. Structurally, they're very small. In fact, they're the smallest cells in the body, about 8 microns in diameter and 2 microns thick. Their kind of most distinguishing feature is they have this kind of indentation. They are kind of caved in on both sides, and that's because in their development, they actually eject their nucleus along with most of the other organelles, and this lack of a cell nucleus in most other organelles maximizes its volume internally so that it can carry more of that oxygen and carbon dioxide. I want to talk about some different proteins associated with our red blood cells. First are spectrin and kind of a familiar friend of ours, actin, which are found on the inner surface of a red blood cell. And I would highlight this. They are what allow for the flexibility and durability of a red blood cell. Very essential, as you can see, because when red blood cells pass through our blood vessels, especially in our capillaries, which are the very smallest of our blood vessels, they need to be able to change shape because they kind of push and shove on each other. So they have to be able to be really kind of flexible and bendy as they do. A lot of those capillaries are just, barely wider than a red blood cell. Now, the lifespan of a red blood cell is only about 120 days, and that's because you can see down there in that picture, they are constantly undergoing wear and tear. Their plasma membranes undergo as they squeeze through the capillaries. But because they have no nucleus and other organelles, or most of the cell organelles, they don't have the ability to kind of replace things when they get damaged and worn out. So that means that, they just kind of will die off, and then that means we have to remove them from the circulation, which is, a normal process because we're constantly producing new red blood cells to replace those that are dying off. Now, within our red blood cells is a very, very strong supply of a protein called hemoglobin. Hemoglobin is very, very prominent in our red blood cells. About 200 and 300 million molecules are found in each cell. And this is the molecule that transports our oxygen and, to a lesser extent, but still importantly, carbon dioxide. Down here at the bottom is an illustration showing you what hemoglobin is made up of, and it's literally heme and globin, which is where the name comes from. The heme is the non-protein part. The globin is the protein part. So hemoglobin is not 100% a protein. It's a protein kind of hybrid. Let's talk about globin. That's what these four, and you can't really tell that there's four of them, but I promise you there are these four blue squiggly lines. There are two what we call beta chains and two alpha chains. The alphas are kind of the lighter kind of blue color, and the betas in this picture are kind of the darker blue. But associated with each of those four chains is the thing that kind of looks like a flying saucer, which is what we're zooming in on and showing you here, the heme. The heme, which contains at the very center of it what we call a ferrous iron, Fe2+, which is where one molecule will bind, or one molecule of oxygen will bind, meaning that one binds here, 2, 3, and then a fourth. So each hemoglobin molecule, as it states here, can transport 4 oxygen molecules. So you take 4 because there's 4 per hemoglobin. You could take 4 times 200 to 300 million. So you're talking about, right around 1 billion oxygen molecules can be carried by one red blood cell. And there are probably trillions of red blood cells in our body, meaning that we have a very, very high capability of carrying oxygen in our red blood cells. Hemoglobin also is involved in regulating blood flow and blood pressure via a release of a gaseous hormone called nitric oxide, which it doesn't actually produce but can bond and carry. And that hormone causes vasodilation, which means that blood vessels will expand and allow more blood to flow through, improving blood flow and enhancing oxygen delivery to various tissues. There's a final protein I want to talk about. It's an enzyme called carbonic anhydrase, which catalyzes, so that means speeds up a reaction joining carbon dioxide and water, forming what's called carbonic acid, which is then going to dissociate or break up into bicarbonate ions, which are essential for maintaining normal blood pH levels. And we already talked about how maintaining pH is a function of our blood. Here's an example of how we do so. We won't really get into any more of that in this class. That's more physiology-based stuff. Okay, on to our white blood cells. The 5 of them are grouped into 2 different categories. The first 3 here that you're writing down, neutrophils, basophils, and eosinophils, are what we call the granulocytes. And then the other 2 are what we call the agranulocytes. Which include lymphocytes and then monocytes. All right. Before we talk about the difference between granulocytes and agranulocytes, let me just talk about white blood cells as a whole. So we know already the other term for them are leukocytes, formed elements in our blood that provide protection against infectious microorganisms and other pathogens. They're different than red blood cells in that they retain their organelles throughout their life, and they're showing much more complex internal structure. You can see here on our pictures, these are zoomed in under a microscope 1,600 times. They do not contain hemoglobin, so they have no role in carrying oxygen or carbon dioxide. Okay. Now, their classification is once again either into the category of granulocytes or agranulocytes. It's all due to either the presence or absence of granules, which are these little kind of dots that you see sprinkled in these first three, but you don't really see so much over here with these two. Either they look grainy or they don't, basically, and it's all due to the organelles, especially the lysosomes and other membrane-bound organelles that show up as these little speckles when you stain them and look at them under a microscope. Okay. Granulocytes, think of them as granulocytes. They look grainy. Other feature of them is that the nucleus is typically kind of irregularly shaped and has some lobes to it. Which I'll talk about that here with each of these. The most prominent of all of our white blood cells are our neutrophils, which kind of have an S or a C shape. You kind of see the S shape to it, or maybe a little bit of a C shape. They are going to contain a nucleus with 3 to 6 lobes. Granules are small and poorly absorb both basic acid and basic and acid stains. Not that that's important. What I would definitely highlight is they are the first leukocytes to arrive at an infection site, responding by chemotaxis to chemicals released by damaged cells. Also highlight that they are important phagocytes, which I always tell students, think of phagocytes like Pac-Man cells, running around chewing stuff up, digesting them. And so they have the ability to like engulf bacteria and kill them. And it's kind of a cool thing. Speaking of, they do and are important in destroying bacteria with their several chemicals. After performing phagocytosis, neutrophils are usually destroyed by antibiotic proteins, forming what we call pus, which is going to also contain other dead tissues. Pus is something we all know, that kind of white gross stuff. You know, we see it when we like pop a zit, or you know, when we have really bad infections, that kind of stuff. Eosinophils, very small number, 2 to 4%, contain a bilobe nucleus. Bi, once again, means 2, so there's 2 lobes, and those are connected by narrow strands of chromatin. Granules stain red with acid dyes and contain digestive enzymes, not super important. What is important is what they do. They combat the effects of histamine, a chemical that we produce in allergic reactions. They also are phagocytizers. Phagocytizing complexes formed by the action of antibodies on antigens, which are just fancy word, or is a fancy word for foreign substances. Although they are not one of our most important phagocytes, they also have the ability to destroy parasitic worms. Our final granulocyte are the basophils that are shown here in letter C. By far and away the least numerous of all of our white blood cells, only 0.5 to 1%, contain a U or S-shaped nucleus with 2 to 5 lobes connected by strands of chromatin. Resemble mast cells, which are found only in connective tissue, and they have granules containing histamine, serotonin, and heparin, chemicals that are released in response to tissue damage and pathogen invasion. Now, we've mentioned this before, but if you put the letter A in front of a word, it means like the opposite or not. So agranulocytes are not grainy in appearance because they just don't have as many, or their granules are completely absent. And unlike these that had bilobed or even 6-lobed nuclei, these are not typically lobed. So let's talk about lymphocytes. About 20 to 25%, often classified as small, medium, or large, contain a circular nucleus, and that nucleus is basically going to occupy the entirety of the plasma membrane. If I zoom in, that's the purple here in our picture. There's just a very little bit of cytoplasm surrounding it, so the nucleus makes up a big portion of the cell. Something very important is that they're the only white blood cells to return to our bloodstream. They are actually also considered to be our major soldiers of the entire immune system, which is the system that helps us, protect against microbes and bacteria and viruses and that kind of stuff. There are 3 types The two that are kind of the most prominent, most important are the what we call T lymphocytes or T cells, and then the B cells or B lymphocytes. Kind of easy. T, think thymus, which is where they mature. B, think bone marrow. B for bone marrow, which is where those mature. Now, all of our blood cells, and we'll talk about this on the next page, originate in the red bone marrow. Okay, but T cells travel to the thymus where they then mature. B cells stay in the bone marrow and mature after they develop in our bone marrow. The third type are natural killer cells, which sound really cool, but really aren't as important for immune purposes as the other two. T cells, what they do is attack aberrant cells. Aberrant is just a fancy word for like abnormal cells, like tumor cells, organ transplant cells, and cells that have become infected by viruses. B cells respond to circulating antigens, things like toxins, viruses, and bacteria, by dividing to produce plasma cells, which then produce antibodies. So B cells end up producing antibodies, but they don't directly do so. B cells become these cells, which then produce the antibodies. And antibodies are the cells that are not the cells, but they're the proteins that then, produce the immunological response. And then natural killer cells attack a wide variety of infectious microbes, as well as certain spontaneously arising tumor cells. Our final type of cells here are about 3 to 8% of our total cells. Monocytes kind of have a kidney bean shaped nucleus surrounded by a large amount of blue-gray staining cytoplasm. Monocytes themselves are not so important, but what is important is what they become, what we call macrophages. Macrophages are formed when monocytes leave the blood and then mature and enlarge into, and you can see we have them listed as phagocytic macrophages. These are the major Pac-Man cells of the body. Okay, so macrophages, which form from monocytes, macrophages, and our neutrophils, you can see, are the most important phagocytes, engulfing things like microbes and cell debris, jumping stuff up like Pac-Man does in the video game. Okay, our final formed element of our blood is our platelets or thrombocytes, which I've already mentioned are not actually cells, but are small fragments of a much larger cell. Marrow cells called megakaryocytes. Here we show one. Only represent about 1 to 2% of the formed elements of our blood. These little speckles that you can see are coming off of the megakaryocyte. Those are the platelets that leave the bone marrow and enter our blood, where they then function. Each of those little platelets is anucleated, meaning it does not have a nucleus, and it's really just going to contain a few organelles surrounded by the plasma membrane. Although they're small and not prominent in number, they are very important. They do things like stop blood loss from damaged blood vessels by promoting the formation of what's called a platelet plug, which is a temporary seal when a blood vessel is broken open. They also have granules that contain chemicals that, once released, promote further blood clotting. Among many other things, there's several things that they actually do I won't really get into. They have about a lifespan of about a week before needing to be removed by macrophages in our spleen and liver. But even though they're only about 5 to 9 days in length, as far as their life goes, we're producing more of them all the time, which is something that kind of relates to what I'm talking about here on this final page, where I introduce hemo or hematopoiesis, two fancy words that mean the same thing, the production of our blood cells. All those blood cells that we just talked about, not just red blood cells or platelets or white blood cells. Is there anything after red bone marrow to talk about? Or write down, I mean? Okay. Although some lymphocytes have a lifetime measured in years, most formed elements of our blood only last hours, days, or weeks and must continually be replaced. This process that we do so is called hemo or hematopoiesis. Both of those terms mean the same thing. Before birth, and we're talking about really early in fetal development, embryonically, we use what's called the yolk sac to perform hemo or hematopoiesis. This is before we have any organs, and once organs develop, it then takes place in places like the liver, the spleen, the thymus, and the lymph nodes. But in just before birth, the last trimester or so, our red bone marrow becomes a primary site, and I would highlight this is the source of our blood cells throughout the entirety of our life. We've talked about red bone marrow before. We learned that it's highly vascularized and that it contains a lot of blood vessels in it, found in the spaces between the trabeculae of our spongy bone. Remember, the trabeculae are the little flakes of our spongy bone. We find red bone marrow in a good portion of our axial skeleton, things like our skull bones and our vertebrae, the pectoral and pelvic girdles, so like our hip bones, proximal epiphyses of the humerus and femur, so the ends of the humerus and femur up towards the shoulder and the hip. Within our red bone marrow are pluripotent stem cells, which are known as hemocytoblasts, that have the capacity to develop into many different types of cells, which reproduce themselves, proliferate, differentiate, as you can see, into a lot of different types of cells. Okay, all those different cells that we just talked about, red blood cells, platelets, all the different white blood cells, plasma cells that come from the B lymphocytes that end up becoming or producing antibodies, macrophages, the Pac-Man cells formed from monocytes, mast cells, which are a type of other cell that are produced originally from that original stem cell. Mast cells are found in areolar connective tissue involved in inflammation. We also can produce from that original cell reticular cells, which are found in our lymph nodes, producing reticular fibers. Adipocytes, which we learned earlier, are fast cells. Osteoblasts, which are formed or are the bone producing or bone building cells in our bones. Chondroblasts, which are found in cartilage, and even muscle cells. Once our blood cells are produced in the marrow, they enter our bloodstream through what we call sinusoids, which are capillaries that look like Swiss cheese because they have these large holes or sinuses in them where they then can enter the blood and mature and begin functioning as part of what they do. Okay, any questions on